Hello students, this is Dr. Faizan Mirza. I'm discussing in this video the questions from LAC operon, and I'll be discussing the question which takes into consideration multiple alleles as well as epsitasis. So just stay with me. Okay, so this question came in October-November 2018, variant 42. Lac operon was shown to you. Lac operon is a section of DNA present in the genome of a bacterium, Escherichia coli. The structural genes of lac operon are fully expressed when E. coli is exposed to high lactose concentration. We have learned this fact in inherited gene chapter. Figure 3.1 is a diagram showing lac operon in a nearby region of E. coli genome. So you can see this is the transcription occurring for all these genes here. Lac A is given to you. So if this is lac A, you can just go back. Lac A, what's present before lac A? We have lac Y here, and before that we have lac Z, and before that we have operator region, and before that we have promoter region. So I have mentioned P as promoter, O as operator, Z as lac Z, and Y as lac A. Lac Y as lac Y. Lac A is already given as a hint to us. So we know that if these undergo transcription, this will cause synthesis of those proteins which the cell needs for digesting lactose. And transcription of the gene I, this produces lac repressor protein. You can recall lac repressor protein. The lac repressor protein will go and the lac repressor protein will go and bind to the operator region. When it is attached to the operator region, the promoter will not be switched on and the transcription of these gene will be stopped. So if you want the lac Z, lac Y, lac A to undergo transcription, you must allow the lac repressor protein to remove from the operator region. So when it's removed from the operator region is free. Now on the promoter region, the RNA polymerase will come and RNA polymerase will start to just bind to it. And because of this, the three genes here, the three structural genes will undergo transcription and mRNA will be made. So they're asking you to name, this is just recalling what we already know. So name the sections 1, 2, 3, and 4. So we know 1, 2, 3, and 4, promoter, operator, lac Z, lac Y, these can be stated. They're saying certain enzymes need to be present inside E. coli for it to be able to take up and use lactose. When the genes of lac operon are expressed, the enzyme beta galactosidase and lactose permease are produced in large quantities. So how is it produced in large quantities? Outline the function of beta galactosidase and lactose permease. So beta galactosidase is the gene that ends up producing the enzyme that helps in the lactose disaccharide to be broken down into simpler sugars, glucose and fructose. So this is the enzyme that break the lactose. Lactose permease, on the other hand, it makes the membrane more of bacteria more permeable to lactose, so lactose can enter much rapidly into the cell. They are saying gene I is located a short distance away from the lac operon. The product of the gene I is a repressive protein, a constitutive protein. So you need to recall what's a repressive protein and what's a constitutive protein. State what is meant by a term constitutive protein. So constitutive proteins is a gene product that is always required by the cell. It's especially essential for cell function. You need to just recall this fact and state it. Explain why the structural genes of lac operon are not expressed when lactose is absent. So when the lactose is absent, the gene I code for the lac repressor protein that binds to the operator region. We know when it's attached to the operator region of the lac operon, when it is bound to there, the RNA polymerase cannot attach to the promoter region of the lac operon. Hence, the structural genes here will not be transcribed and into mRNA, and gene expression for the structural genes of lac operon does not occur. So you just need to state that. A strain of E. coli has been produced by the mutation of gene I. Expression of this gene results in a non-functional repressor protein. So there's a negative effect of this mutation that will have on the effect of E. coli. They are saying that now this I is producing a protein which is not binding to operator. So if it doesn't bind there, so promoter is always available for the RNA polymerase to work on. The cell will keep on producing all these three proteins of the lac Z, lac Y, and lac A. And all these proteins are unnecessary. These are all non-essential. This bacteria is not taking lactose. Why is it even need these proteins? So the negative effect would be the structural genes of lac operon will be transcribed, resulting in substantial loss of ATP as well as amino acid, because this is the loss that actually the cell will have to incur. ATP will be lost and amino acid will be lost because the proteins which are being made are non-essential for this particular cell anymore. Another question of lac operon, I'll not go into the details because it's exactly the same as the previous one. So let's come to another question here of epsitasis. This saying there, this is a flower of common morning glory plant having several different phenotypes. This name is given to you. You know the first name here represents the genus. The second name here represents the species of this. So this is how we use the binomial nomenclature. So furthermore, you can see this is the flower and this is the eye in the center of the flower. This is what actually they are mentioning. Flower color eye If our Clark in this particular species controlled by two genes on two different chromosomes, gene R and small r, which code for a protein involved in pigment production, has two alleles. So this is the pigment producing allele. The dominant R allow pigment production and small r prevents pigment production, resulting in the white flower. So you just annotate here that white flower will always have a small r, irrespective of whatever is there. If small r is there, the flower will be white. Gene T capital T small t, which determines the type of pigment produced, has two alleles. The dominant allele capital T shows purple flowers and small t results in red flowers. So whenever there's a small t, the flower color would actually be resulting in red flowers. Define the term alleles. So alleles are the different variants of the same gene. They differ in nucleotide sequence and end up producing a variant of the polypeptide. They always occupy the same position on the same. They always occupy the same gene locus on homologous chromosomes. So you can state these points to get the common marks here. So just the ways in which expression of allele R allows pigment production. So how it allows pigment production? What is the effect? So you probably do not know that. So how you work out? You just need to suggest. So probably this is an example of gene interaction. The allele R may code for an enzyme that in pigment production, or it may block an inhibitor of pigment allele. It may behave as a transcription factor. We know transcription factors are all proteins. Keep that in mind. All transcription factors are proteins, whereas the promoter sequence on which the transcription factor bind, that promoter sequence is actually a DNA sequence. It's the DNA sequence present on the chromosome just beside the gene, and the transcription factors go and attach to the DNA sequence on the promoter, and when they are attached, gene expression. So I have covered this in various questions previously and in my other videos as well. So I really hope that this concept is not a contradicting concept anymore. Furthermore, they're saying that you need to just complete this complete figure 3.2 to show the results of a cross between these two plants, heterozygous at both loci. So you just cross them, heterozygous at both loci, they will have the heterozygous condition. So when you are having heterozygous for both, you just need to break this in such a way that capital R and small r will go in separate gametes. So you will have a capital R in one and a small r in the other. Capital R, small r cannot be in the same gamete at all. So capital R can go with a capital T, capital R can go with a small t. Similarly, small r can go with a capital T, small r can go with a capital with a small t and capital T either. So this means that this is independent assortment. Whenever all three of these are present, we can expect that this is actually independent assortment occurring. And when independent assortment is occurring, this probably means that there is no linkage. So you just need to just recall this as well. And we know that when there is no linkage occurring, we get the ratio of 9331, and we have 4 different phenotypes occurring, out of which 2 are the recombinants. So we go for the same cross here, we add the gametes here, and we go for the crosses here. But when we come across, what do we see when we start to write we start to write their phenotypes here. For example, if RT and RT is present here, capital R and capital T, so this product will be capital R, capital R, capital T, capital T. Similarly, the other will be capital R, capital T, small t, when you are crossing them together. Why is this capital R, small r? Because this capital R, small r coming from capital R and small r, and this capital T, small t comes from this capital T and small t. So this is how it comes. So when we are writing the pigments here, what are we doing? We'll just look for wherever we have small r. So we can just go back. We know small r are white flowers. So wherever we have small r's, we'll just mention white. So all these 4 are white, whereas others are non-white. So out of all those others which are non-white, we need to state which one is having a purple color and which is having a red color. We can recall the red colors whenever there is small t present. So small t will be red. So this will be red, small t, and small t will be red, small t will be red. But this small t cannot be red because there is small r present. We know that it is mentioned already in the question. The recessive allele R prevents production of any color. So when their small r's are present together. So when you start to count them, the count comes out to be 9, 3, and 4. So 9 purple flowers, you have 9 purple, you have 3 red, and 4 white. This is because this is an example of interaction between loci. This is epsitasis occurring. So this ratio is not 9331 actually. Why is it not 9331? Because although independent assortment is occurring, the interaction between loci doesn't allow the 9331 ratio to occur. The ratio is 934. Although the reason is not mentioned, the reason is not asked, but they may ask you the reason that why this is an example of interaction between loci. Because if it was not there was no interaction between loci, the ratio would have been 9331, and there would have been 4 different phenotypes. Here we have 3 different phenotypes occurring, and this is because there is interaction and epsitasis occurring between loci. This is the simplest question. I think let's come to another question here. This is October November 2019 variant 41. Initially, they have asked you just to outline the outline. This is state and tick across for meiosis and mitosis. I think this case is something very simple. Chromosome number is maintained in meiosis. It's not in mitosis. It is homologous chromosomes pair up in meiosis, but not in mitosis. Sister chromatids separate in meiosis and they separate in mitosis as well. Occur in prokaryotes? No, no. For both, you need to recall that prokaryotes divide by binary fission and not by mitosis. So binary fission is the process by which they divide, and mitosis and meiosis only occur in cells that have nucleus because they both are the type of nuclear division. Now that we have stated that, we move on to compare the role of meiosis and gametes response cells, and this is not a part of syllabus anymore. We can skip this, the syllabus of 2022. So you can just go to the syllabus 2022. You will find out that gametogenesis is not a part of syllabus anymore. Flower color is an important sexual reproduction insect plant native plants. In rose periwinkle, this actually flower color is controlled by three genes. There are multiple genes, capital R, small r, capital D, small d, capital P, small p, which interact together. So this is an example of epsitasis as well, and this is an example of multiple alleles as well. So this is a question which is taking two things together, epistasis as well as multiple alleles. So this is the petal of the flower. This is the red eye in the center of the flower. Now, the information given to us is the presence of R, capital R, results in the production of red pigment in the center and the red in the center of the flower, which is called red eye. So if there is capital R dominant presence, so you can say that it will have a red eye. So wherever you have you have a capital R, you can state that organism, that plant will definitely have red eyes. So red eyes, and you can mention red eye in the phenotype because the capital R is there. Furthermore, they're saying capital D allele and capital P allele are only expressed when R allele is present. So for the expression of D and P, R must be there. So B, D, and P will only show their effect in the phenotype if capital R is already there, either as a heterozygous or a homozygous condition. So they are saying when D is present and R is present, the flower has dark pink petals with red eye. So you just need to see where this is present, and you will just mention which one is having pink petals. So this is the furthermore, they are saying when the allele P allele and R allele are present, the flower has pale pink petals with a red eye. When the D allele is present, the P allele and R allele are all present. The flower has dark pink petals with a red eye. The recessive allele RDP prevents pigment production altogether and having white petals and no red eye. So use your analytical skills now. You need to just read very carefully which letter represents what and how these alleles are interacting with one another. Use your analytical skills with the given information and just apply. So we know that with the RR red, as I've already mentioned to you. So when this is capital D and P present together with the capital R, so dark pink and pale will be there. So we know that dark pink petals will be there and red eyes will be there. So apart from that, if a smaller is present, then definitely it'll be white petals. And D and P are both present, but since the R is not there, so they will have no red eyes in the white center. So the same goes for the D, small p. So this will cause the white petals and red eye. Why? Because capital R is present there. So just use your analytical skills to just mention the phenotypes here. They are saying the pigment causing flower color in the rose periwinkle are found by biosynthetic pathway. The allele are mutated to produce small r allele. The R allele codes for non-functional protein. Explain how the mutation that changes R to R results in non-pigment production, the synthesis in the rose periwinkle. So the mutation could have been due to change in the codon due to base substitution, base deletion, or base addition, where addition results in frame shift. We know that frame shift is because of addition and deletion. The outcome of such mutations could be that that the another amino acid gets added to the growing chain. This is missense mutation. The primary structure would be changed and the 3D shape would be changed. Just recall what you learned in AS, and because that information is relevant for you, the active site would not allow the active substrate to the actual substrate to attach to it, and the biosynthetic pathway will start to stop. One mutation that changes the flower color of rose periwinkle occurs in the region of DNA that does not code for a polypeptide. Recall AS. Which part of the DNA or chromosome actually doesn't code for a polypeptide? So the promoter region doesn't code for it. The telomere doesn't code for it. So you can mention either any other of these. So these are the binding site for the enzyme. So you can just mention it here. I hope these questions were helpful for you. That's it from my side. Thank you so much.